the archived session on our website as well, so people can continue to, to visit at a later point that's more convenient to them.
Yeah,
description so they understand what's there, but no, that's a good point.
Yeah, have both up there so you can listen to it if you want to. Or maybe somebody's just interested in one particular slide, they can download that one.
Yeah, that's a good point. So I'll ask Rhonda to do that. I'm pretty sure I have PDF copies of all the previous ones, so we can do that for the last 3 that we've held. So I think that's pretty much it. So it looks like the technology is working well. And so we'll just hang on for 10 minutes or so. You can catch up on some work. And then I'll start it, probably not promptly at noon, but a few minutes after, just to give a few more people to join.
Oh, Anita, we got to talk about doing the metabolomics, because I want to switch to a MS rather than NMR.
Okay, which is a different platform. Is it? Is that when you were hoping to do some trial run?
Yeah. So I've done some trials, and it's going to be way better.
Okay. Do you have a sense of how much funding you'll need to do, and for how many specimens you plan on doing it on?
Yeah, so that was part of this. So I've got them done. It's going to cost me maybe about $52 a sample.
Okay.
But now the analysis time is going to be way less.
Less, okay.
Way, way less.
And do you recall what you had originally projected as part of the
Well,
budget?
originally we were going to do the NMR, which was $35 a sample, but then we have to take into account the fact that it takes 1 hour to do a spectral analysis. So if you're doing 1,000 samples, that's 1,000 hours, and that's before you get to the bioinformatic analysis.
Okay.
So I'll just write out the reasons why I want to switch, and it's going to be. I
think that's fair. I know that Mike Surratt has also been looking at updating some of the technology that he would be using for his microbiome analysis, because again, he's been able to, bring down the cost and it actually get a lot more information than what was in the original proposal. So I think that
technology has definitely improved dramatically since we wrote the original. So
I don't think there was a problem. I remember seeing that in your email, Karen. I apologize for not responding, but if you just want to put together a brief little proposal on how many samples you hope to do this on, just to make sure that for quality control or for whatever other purposes you'd want to do a bit of a trial run.
Yeah, I will do that.
Okay, perfect. And then I did set, because actually when I was putting together our projections to CIHR, I did sort of project an earlier expense associated to some of the analytics for that same purpose. So I think it's, we've just reallocated. I didn't necessarily increase the budget, but I just did some reallocation. So instead of spending it all at the end when we do all the analytics, we would do sort of a bit of a test run.
Okay, sounds good. We'll talk later about this then.
Yeah, absolutely.
Okay.
Okay, so
Oh, okay.
Mm-hmm.
Hello, everyone. We're going to get started in just about another minute or two, just to give people a little bit more chance to sign in here. But if you haven't already done so, please just set yourself to mute. That will help us with the AV here. Thank you. Okay, so welcome everybody. Thank you for joining us in this webinar series. This is obviously one of monthly series that the Imagine Network puts together to provide training and capacity building for the research community. Not just involved in Imagine, but obviously interested in the research mandate that we're involved with. So my name is Ida Fernandez. For those that don't know me, I'm the Executive Director for the Imagine Network. We do hold these monthly webinar series as part of our capacity building core program. And we're delighted that this month we have Dr. Karen Madsen presenting to you today on how to optimize metabolomics in your clinical research study. And Karen, next slide. So just a little bit about the Imagine Network. So the Imagine Network is investigating the interactions between inflammation, microbiome, diet, and mental health in patients with IBD and IBS. And our mission is to transform the management of these diseases and their associated mental health issues. So next slide, Karen. So our speaker today, as I mentioned, is Dr. Karen Madsen from the University of Alberta. Karen is our lead for metabolomics within the Imagine Network. Dr. Madsen completed her BSci and Master's in Science in Biochemistry at the University of Manitoba, and then did her PhD in GI Physiology at University of Calgary. She's leading researcher in intestinal impermeability and gut microbiota and their effects on disease and health. She's co-authored 250 publications and abstracts. She holds a number of research grants and awards as well. So please join me in welcoming Dr. Karen Madsen, who will present today. Just as a little bit of a heads up, if you have questions, we've reserved some time for Q&A at the end of the session. So please just enter them into the chat box, and you'll see a little icon at the bottom of your screen that says chat. Just type your questions directly in there. And at the end of her talk, I will moderate and ask your questions for you. So please hang on. Also note that we will archive, we're recording the session, and we'll archive it on the Imagine website for future viewing. So you'll have an opportunity to revisit this, this webinar session at a later point. So thank you all again for joining, and now passing it over to Dr. Madsen.
Hey, thanks, Ada. Welcome everybody for joining to talk about metabolomics. So what I want to really kind of go over is how to incorporate metabolomic analysis into human studies. And for you to gain an appreciation of the different types of platforms that are used for these studies, as well as some of the challenges and limitations associated with these types of studies. So first, what is a metabolome? It's simply all small molecules found in a biological or environmental sample. These can be endogenous, naturally produced by the host. They can be exogenous, coming from drugs, toxins, dietary compounds. And there's a whole host of metabolites that are produced by microbes within us. They can be modified by us. And so you end up with these microbe host co-metabolites, which are very interesting compounds. Now, if you look at the metabolome, starting, with genes, we have 4 nucleotide bases, got about 20,000 genes. If we look at the proteins, about 20 amino acids, about 600,000 known proteins. When we get into metabolites, you're looking at over 3,000 chemical classes of compounds. You're looking at 1 million different metabolites. And the metabolome is really a measurement of what is happening to you right now. It's your phenotype. And it's very sensitive to both environmental influences as well as physiological influences. Okay. So it changes all the time. It is what is happening to you now. And if we look at the interactions between us and our microbes, so we have all of these metabolites that we are producing all the time. We eat food, so there is a dietary food metabolome. These compounds that are found in the diet are acted upon by microbes as they pass through the gut, and they're acted upon by us, the host. There's also a drug metabolome. There's also toxins, environmental pollution metabolome, and all of these are linked together with the microbiome. Now, one thing you have to remember is that the time scale for metabolite changes is very different. So when we eat something or we do something, your genes don't change. Proteins will change slower, but if you look at metabolites, there are dramatic shifts, changes, up to the minute changes, very, very fast changes in a whole host of metabolites. So what can we use metabolomics for? Precision diagnostics, looking at how patients respond to different drugs. Patient stratification, maybe looking at different subtypes of phenotypes. Maybe you're looking for a biomarker, and we'll talk about those. Maybe you're looking to see if you can identify early disease in a patient before you have more clinical signs. Maybe you're looking for molecular pathways that are underlying health states or disease states. Maybe you're looking for host response to environmental exposure. Or maybe you're interested in looking at microbiome and immune profiling. So all of these can be done by metabolomics. You've got all of these factors, genetic factors, age, the environment, drugs, nutrition, lifestyle, all of these are affecting the phenotype. So obviously, the first thing you have to do is define your objective. Are you looking for general information to help you form a hypothesis? You just want to see what's there. Do you have a hypothesis that you want to validate? Or maybe you want to discover biomarkers. Maybe you want to have a predictive biomarker. So you want to determine who might respond to a treatment. Maybe you want to use a diagnostic biomarker. So maybe you want to be able to take a urine sample and see if that person has irritable bowel syndrome or inflammatory bowel disease. Maybe you want to use a prognostic biomarker to determine patient outcomes. So maybe you want to see if there's a particular biomarker that this patient will go on to have very serious disease, or maybe this patient will stay in remission. Or maybe you're interested in drugs and the host and interactions. So maybe you want to look at pharmacodynamic biomarkers. Maybe you just want to gain an understanding of the pathophysiology of disease. Maybe you want to identify new drug targets, or maybe you want to try to design new treatments. So depending on what your objective is, it will determine how and what kind of analysis you will do. So when we're talking about human studies, the main ones that are used are either cross-sectional, where you take people with the disease versus a matched group of people without the disease. Usually at one time point, you take a sample and you see if they're different. Or you can do either observational, so you just take samples from a patient, follow them, their natural course of their disease, or see who develops disease over a long period of time, and you take samples periodically. Or you can do a longitudinal, where you do pre and post intervention, where you'll take a baseline sample, you'll give them a drug or a nutritional therapy, and then you'll take samples after the intervention. And your goal, hopefully, is to get one of these beautiful PCA plots where you have maybe a case and a control, but they're starting to separate and you get differences, and then you can look into what is driving this difference. Whatever you choose to do for your populations, you are going to end up with massive data sets. You're going to have to do a lot of multivariate stats, and you're going to have to use a lot of different clustering and classification techniques such as PCA or PLSDA in order to understand what is going on. Okay, so let's talk about designing your study. Okay, you're going to have your question. What is it? Is it hypothesis driven, or are you looking to generate hypothesis? And then we get into the sample size calculations, which everybody asks us about. These are really difficult to do. When you're doing metabolomics, you can look in the literature, have any small pilot studies been done to point you in a direction that you may be able to see from that what is changing and how much of a degree of change you're seeing. Lots of times this is not available in the literature. Maybe you can do your own pilot study, maybe not. Maybe if you're doing a clinical study, you can use metabolomics as a secondary outcome and have a primary outcome with something that you do have a hard outcome for. What kind of controls are you going to be able to use? What kind of samples can you obtain? Can you obtain blood? Can you obtain urine? How much sample can you obtain? What about preparation and storage? What about the logistics of this? How much control do you really have over external variables? So the problem with metabolomics is that everything affects it. So do you have any control over their diet? Do you have any control over their environmental exposure? Usually not. So the more information you collect about the people in your study and the samples you're collecting, the better. Because you can use this information in your multivariate analysis later on down the road. You have to think about your requirements for data storage, bioinformatics, and computing capabilities. Who is going to do your bioinformatics? We'll come back to that. What about the number of biological and technical replicates can you do? And a lot of times this comes down to your financial resources, because metabolomics is not cheap. Depending on what kind of analysis, you can go anywhere from $100 to $1,000 a sample. This obviously affects how many technical replicates you are able to do. Okay, what about which sample to choose? You can take blood, serum, plasma, very easy to get. You can also use urine, sweat, saliva, or stool. Whichever one you choose, the metabolome profile is going to be unique to that sample. Okay, so a urine profile is very different from a blood profile. All of them will vary depending upon the time of day, the diet, age, sex, and other variables. So it's interesting if you compare urine and blood. Urine is easier to get. It's less invasive. Nobody really minds giving a urine sample. And everything that is in blood is in urine. But the concentrations will differ. So you may have a metabolite in blood that is there at such a low level that it's non-detectable. But because the kidney can concentrate, and it does concentrate a lot of things, you will be able to pick it up in urine, whereas you couldn't pick it up in blood. So concentrations are different. These all represent what's going on in the body. Same with these. Sample collection must be standardized. We cannot emphasize this enough. This is so important in a human study. Biofluids are active. Many things display circadian rhythms that you find in blood. So you have to standardize the time of collection. Urine, it's best to get an early morning before eating urine. It can be stored at 4 degrees for up to 4 hours without a problem. So you can say have somebody collect it at home, put it in the fridge and then bring it in, keeping it on ice. For blood, the time between collection and processing should not exceed 2 hours. Stool, you really can't produce stool on demand. So usually you have to get people to collect it themselves. It can be frozen and brought frozen, or it can be placed in a stabilization buffer, which is pretty much ethanol, for up to 7 days at room temperature. OmniGut DNA Genotech has now developed a specific stabilization buffer for metabolomics. So again, it's a very easy, it's called OmniMetGut. So it comes in a tube, patients just collect a little bit of stool, put it in, it can stay at room temperature for 7 days, no problem. So that makes it really easy to collect and bring in or send in. Urine, it has to go through centrifugation and filtration to remove cells prior to freezing. So this has to be done. Think, where are the samples being collected from? Are they being collected at clinic visits? Are patients bringing in the samples? And where will samples be stored? So you have to think about all of these logistics. You also want to limit freeze thawing. So absolutely freezing in aliquots. So don't just get a big thing of urine and freeze it in that one big thing. Put it into aliquots because you don't want to do a lot of freeze thawing. Preferably at -80. And then the extractions following that are totally dependent upon what kind of analysis and instrument you're going to be using. So I just put up project planning with all of the things that you need to think about when you are planning your clinical trial. So the questions related to the design of the experiment, the research question, what kind of stats you're going to be doing, where you're going to keep your samples, so biobanking plan, the number and types of samples you're going to be collecting, the number of aliquots you're going to be saving. I'm looking at where the sample is coming. Is it coming from home, coming from a single center, coming from multiple centers? If you're doing at multiple centers, you have to make sure you have standard operating procedures that all centers follow. This is really, really important that it is standardized across all centers. Or you're going to end up with so much noise in your system, you will not be able to understand what is going on. Again, looking at exact collection methods, what you're doing, what time of day you're going to be collecting. Blood is a little bit less problems related to the time of day as is urine. You need to know though, is it a fasting state or a fed state that needs to be written down? If you're taking multiple tissues, you need to specify what order those tissues are taken in. For processing, you need to really think about things like who is going to do it, where is it going to be done? If samples, say, are collected in the clinic, do you have a centrifuge in the clinic that the samples can be centrifuged and filtered? Do you have a freezer in the clinic that they can then be put in the freezer? Or do you have to run these samples over to another building? And who's going to do that? So these are just all things that when you're setting up the study, you need really to think about and put in place. And then for transport and storage, if they're collecting at home, how are they going to get to the center? Ice pack, again, you have to think about this and figure out the best way to do that. Okay, so that's a little bit about sample collection, really important. What about now getting into measuring the metabolome? So basically you have your sample, you extract, you homogenize tissue, extract a fluid, do your chemical analysis, and then do your data analysis. Simple. So what kind of studies can you do? Okay, they can range from targeted compound analysis. So this is where you take a sample and you are doing a quantification of specific metabolites of interest. So say you're wanting to look at bile acids. So you basically have a analysis of just bile acids and they're all quantified. Then you can do metabolite profiling, which is a little bit beyond specific metabolites, where you're looking at qualitatively or quantitatively a group of related compounds associated with specific metabolic profiles. So maybe you're looking at energy, or maybe you're looking at tryptophan metabolism and all the metabolites associated with that. Then you're getting into metabolomics, which is looking at a totally comprehensive analysis of the whole metabolome under a given set of conditions. Then there's also fingerprinting, which you're not really looking at the identification of individual compounds, but really you're looking at the comparison of patterns in specific diseases or how a sample changes in response to some factor. So again, a fingerprint rather than specific metabolites. And then we also talk about targeted versus untargeted metabolomics. So targeted really means you're looking for specific metabolites. It's quantifiable. You use quality controls, internal standards when you're running the analysis, and you end up with concentrations, quantities. Untargeted, you don't have any type of internal controls. You are looking at what kind of peaks. You have to analyze the peaks, and then you have to do an analysis. So a targeted metabolomics workflow kind of looks like this. You run your samples, you get your peaks, you're using hits with internal controls, and it's a relatively routine quantification. If you're doing biomarker discovery, you absolutely have to have quantification because you need to see what normal and abnormal values are for diagnosis prognostic purposes. These are often kit-based. BioCrates has a lot of different kits. They could range from their largest one, which is the Quant 500, which is up to 630 metabolites from 26 different biochemical classes. It's in a 96 well plate. It has calibration standards, quality controls, and test samples. And they have things that are very focused. They have one for bile acids where you do 20 bile acids. And they have a new one for tryptophan metabolism where they have 70 metabolites from the tryptophan pathway. So getting a huge range of different kits with the numbers and specific things you are looking for. Centers also have their own in-house kits for targeted different workflows. Untargeted is very different. So in this workflow, you take your samples, you do a data acquisition using various different platforms. You'll get all of these peaks. You have to do a lot of work looking at extracting these peaks. You then do a compound identification off the spectral profile that you get, looking at a lot of different databases. You do a lot of stats looking at univariate, multivariate types of analysis. And then you have to do your pathway analysis, interpretation, and looking at biological models. If we compare targeted versus untargeted, so targeted is limited, obviously, but again, you can go up to 600 different metabolites, even in a targeted format. But you are limited in potential for discovering new compounds. It is mainly hypothesis driven because you have an idea of what areas you're interested in. You do get a precise concentration measurement. It's easily automated. It's for high throughput, highly standardized. Untargeted, you do get a lot larger coverage. You can discover novel and often unknown compounds. It is hypothesis generating, but you do not get a concentration measurement. It's definitely not automated, and it is definitely not fast, and it requires much more standardization. So one thing to remember when you're measuring metabolome is that there is not one single platform that can measure the entire metabolome in any given sample. So usually NMR or mass specs are used. They're usually coupled with separation techniques, such as gas chromatography, liquid chromatography, ion mobility systems, or you can combine them. So what happens is you take your sample, you put it through a, let's say, liquid chromatography to separate the sample, and then it goes into the mass spec for analysis. There is also an LC NMR method that's being looked at. And the technology and sensitivity is different. So NMR has the lowest sensitivity, looking the micromolar, followed by GC, followed by LCMS or CEMS. So a lot of people are looking at comparing NMR and mass spec when they're trying to choose which one to use. So NMR separates signals by their resonance frequency in a magnetic field, detects all organic classes, uses a small amount of sample. It's highly reproducible. It requires little sample preparation, goes directly onto the instrument. It's non-destructive. So that means you can run your sample through an NMR, take your sample, and then you could run it through mass spec if you wanted to, or run it through ELISA. You can use it for anything. It's better for structure, less batch effects, but lower sensitivity, more time required for analysis, so it's less automated. So once you get your peaks, you have to spend more time manually looking at the peaks and trying to deconvolute them. And it cannot detect inorganic ions or salts. So mass spec is a mass to charge ratio, smaller amount of sample, So if you use 100 microliters for NMR, you can get away with 10 microliters for mass spec. Definitely a higher sensitivity, usually paired with chromatography. Less time required for analysis, but it is less reproducible. It destroys the sample, so you can't use it for anything else. And because it definitely has more batch effects if you're looking at a long-term study. Just to give you an idea of what classes of compounds can be detected with the different platforms. NMR, primarily amino acids, alcohols, amines, sugars, organic acids, and water-soluble metabolites. Gas chromatography, MS, more of the amino acids, organic acids, fatty acids, bile acids. And when you get into the liquid chromatography, MS, you get a whole host of compounds to the nanomolar concentration. So this is what you end up with, a chromatogram with all of these peaks, and you have to identify each peak. So you do that through things like the Human Metabolome database. It's an electronic database which links all chemical, clinical, and biochemical data. Over 100,000 metabolites are in the database. It contains water-soluble and lipid-soluble metabolites that are both abundant and relatively rare. And it links a whole host of other databases. There's also a new database now, which is called the Fecal Metabolome database. It contains over 6,000 metabolites right now. It does include known microbial compounds from the E. coli database and the yeast database. It includes data on likely or known species or origin, and it contains a whole host of experimental NMR and MS and GC spectra. So if you're interested in stool metabolomics, this is now available. So what about costs? Everyone wants to know what it costs. Well, this is taken from the Metabolomics Innovation Center here at the U of A. There's a whole lot of commercial places that you can go. But to give you an idea of what the costs can be, for instance, the Metabolomics Center here has the LCDIMS metabolome profiling assay for $60 a sample that will give you 146 different types of metabolites. A quantitative GC-MS for $70. You can do NMR for serum for $50, urine and stool for $125 a sample. But again, there's a whole host of different types of analysis, can range $700 up to $400 a sample. But these costs do not include the bioinformatic analysis that needs to follow. This is basically giving them your sample, and then they will give you back the raw data. So bioinformatics is really the key to success. You're getting hundreds and thousands of metabolites. You have to turn that into meaningful biological insights. So if you're receiving the raw study data, you have to have somebody in your group that can dedicate a lot of time and resources doing the stats and researching the biological implications of these findings. Now, if you don't want to do it yourself, if you don't have somebody who's able to do that, a lot of centers will offer bioinformatic analysis and specialized interpretation tools for a fee. And you can discuss this with them. There's a lot of centers across Canada that offer this kind of service, because most people don't have their own NMR facility, and most people don't own their own mass spec. Okay, just a few applications that we have done to look at. So a study where we were looking at ulcerative colitis patients, and we were interested, could you use metabolomics to predict who is going to have a flare in the future? So we took ulcerative colitis patients in remission, took baseline samples, urine and serum samples, and then we just followed them for 12 months to see who had a disease flare and who stayed in remission. And then we looked back and we did the baseline urine and serum samples. We did both an LCDI-MS, and an NMR analysis. And what we found that here we had, these are the patients that relapsed, these are the patients who stayed in remission. This is what their baseline metabolomic data looked like when we analyzed it. So again, you could see who was going to have a flare. And these are some of the metabolites that popped up. And when you do the analysis of these, it looks like there was possible changes in energy-related metabolic pathways and oxidative damage occurring prior to disease relapse. So again, this is something that was predictive of future disease. Here is another one we did where we looked at Crohn's disease patients. In this case, the patients had ileocecal resection, so they had had surgery. Unfortunately, a lot of patients will relapse within 6 to 12 months following surgery. And we were looking at, could we take a sample and be able to identify those patients that were having a relapse rather than having to undergo colonoscopy. So again, we took urine samples. We did profiling again with a combination of NMR and mass spec. And then we correlated these with clinical findings. So we were able to see, were these patients sick? Did they have a relapse? And what did their metabolomics look like? Again, so the red, these are the patients who relapsed. No, these are the patients who relapsed. These are the patients who stayed in remission. There was a separation, so we could tell by their urine sample, and again, some of the metabolites that were able to discriminate between those suffering disease relapse and those that were not. A study looking at the difference between IBD patients and irritable bowel syndrome patients. Urine samples were collected from 39 IBS and 53 ulcerative colitis patients. Again, combined GC and LC-MS method. And the two populations differed very nicely. And looking at the metabolites that popped up, some of these lactic acid, histidine, proline. You can also use it to analyze response to specific treatments. In this case, we looked at the response to low and high FODMAP diets. So patients with irritable bowel syndrome, we took samples at baseline, and then they were randomized to either a low or high FODMAP diet for 3 weeks. We looked at them again in 3 weeks, and you can see that the people on the different diets are starting to separate. And it was interesting because in this study, histamine was identified as a major metabolite in those patients who responded to the FODMAP diet. And then this is a case that looking at development of a urinary test for colonic polyps. This was initiated by Drs. Richard Fedork and Hallie Wong here at the University of Alberta. So they wanted to develop a urinary test that could basically diagnose patients who had polyps without them having to undergo a colonoscopy. So they simply started off by collecting samples from patients with polyps and patients without polyps. They ran through a lot of NMR spectra, moving over to MS, and they initially came up with a profile that was linked with those patients who had polyps. They refined this, they validated it through thousands of different patients. They used machine learning and algorithms to identify those metabolites that were the most important. They were able to bring it down to about, I believe, to about 12 metabolites. They further validated the test, moved it over to a mass spec for high throughput, and I believe they're down to 3 or 4 different metabolites now that they can use to detect the presence of polyps. So it's a very good non-invasive test that is now being used clinically to diagnose polyps. Okay, some of the challenges and limitations. There is no universal method that has the sensitivity and specificity to identify and quantify all existing metabolites in a sample. Samples contain a huge, vast diversity of different classes that vary on orders of magnitude from picomole up to millimolar. You cannot use one platform to identify anything. Stability to oxygen, light, temperature, turnover rates differ for all compounds. So you may have some compounds that are very sensitive to oxygen or light and some that are not. When we talk about quantification, the accuracy is still challenging because it depends on many factors. How complete is your sample extraction? A lot of errors can be introduced there. Reproducibility of instrument analysis steps and systemic matrix effects. So when you're looking at a sample, you have a lot of compounds that you're trying to extract. You have a lot of compounds that may mask being able to modify or see those particular compounds. There still exists a major barrier to translation of experimental findings to clinical use. The data is highly complex. Reproducibility of results is not that great at this point, simply because of all the different ways people extract, different ways people process, different experimental ways of analyzing. So this remains a major barrier. And of course, the cost and requirement for extensive bioinformatic analysis. These are expensive to do, and you need somebody who really is good at bioinformatics to do the interpretation. Okay, for those of you who are interested in metabolomic studies, there are hubs across Canada. Largest one at the University of Alberta, but also hubs at Calgary, Victoria, McGill, McMaster universities. These are all part of the National Metabolomics Network. If you are interested, you can contact any of these hubs for information. They will help you in designing your study. You tell them what you want to do. They will definitely help you in what platform to choose. They will give you help with every part of your study. They provide a lot of metabolomic services to pathway analysis, to global profiling and comprehensive analysis. And they will link all of the databases for you within these centers. So again, if you are really interested, I really highly recommend that you meet with experts in these centers because they have the knowledge. They can give you specific answers to any questions you have about designing or running or analyzing your data. And I will end there and happy to take any questions.
So thank you so much, Karen, for a wonderful presentation. Very informative. Again, I'll just pause here a few minutes for people to type in their questions in the chat box area, and I will field them. A few are just starting to come in. So again, type away and we'll have Karen address them. So here's a question, Karen. Is one of the reasons for the lack of reproducibility also a multiple testing issue, as well as the various methodological problems of lack of standardization? If so, would it not be better to do split analysis with a test and a validation set?
Yeah, absolutely. I didn't want to get into that, but whenever we're doing a metabolomic study, we try to do, or you should be doing, a test and a validation at the same time when you're doing your analysis. So that means basically you take all of your data, you will split it, you'll do permutation analysis where you run your samples with a subset and then keep running it with different subsets to do your analysis. So absolutely.
Okay. Just another question, a little bit more related to the Imagine study specifically, Karen. in Imagine, obviously, urine is the biosample that's been being collected for metabolomic analysis. Can you explain why specifically, and what advantage did urine provide over some of the other samples that were already being collected, such as blood and stool?
Like I said, you do get different information related to environmental exposures between blood and urine. Obviously, stool is quite different from blood and urine. And one of the things we can get out of urine is, as I alluded, things that you are too low to pick up in blood, you can pick up in urine. So it's good to have both samples. Maybe we'll do metabolomics on selected serum samples or blood samples for selected cohorts. Whereas we will be doing urine metabolomics on most subjects. It's just good to have both samples because it gives you different information, really. Blood is less related to dietary intake. Urine is more sensitive to dietary intake, but they're different. So it's good to have both.
And here's another question. So if the metabolomic profiles are driven by so many different variables, how do you get a solid baseline to compare testing between groups?
Yeah, that is a really good question. That is why you try to control. Obviously, if you're doing case controls, you try to control for sex, age, diet, whatever you can control for. When you're matching cases with controls, the better. You can also collect information about different exposures and put it into multivariate analysis. And that can kind of show you what is driving the separation. It all comes down to really the bioinformatics that you start doing with the data to remove a lot of the noise, a lot of the extraneous factors, and then you add that with all of your meta information that you have collected.
Thank you.
It's not easy.
Clearly. Another question. So this person says, I understand the reason for linking what you find with biological process, but biologic plausibility could be created for anything. And so how do you control for the bias of finding something and then retrofitting a biological process that makes for a good story?
You have to validate. So if you find a signal in one study, one population, that's only one. You have to go out and then you have to validate that in another population. You have to validate that in experimental studies. You take your data and your findings, and then you try to reproduce it and you try to validate it. Okay, you can't just go with one study and say, okay, that's it. You can't do that.
So for maybe for some young researchers here, Karen, I mean, you've provided some important links to different collaborators that can help them with metabolomic analysis. Is that sort of the recommended route if you don't have that in-house expertise? Is simply just to connect in with these services at other institutions?
Absolutely, that's what they're there for, and they are incredibly helpful. Because metabolomics is it's an expertise that not everybody has. So people don't go out and buy their own mass spec and start running their own metabolomics. It's really cost prohibitive to do that. Link up with anybody at any of these hubs. There's also commercial centers. I haven't dealt with them, so I'm not sure how helpful they are, but I mean, they're popping up all over the place that will give you advice and run samples and do whatever it is you want. So they can also be contacted as well.
And this one's a little more forward thinking, Karen, but do you foresee a time when metabolite urine strips will be available to general public that can help monitor or manage various symptoms?
I do, because if you look even at the polyp story that Dr. Fedorik and Wang developed here, I mean, 10 years ago, they started trying to identify if they could see who has polyps. They went through thousands of tests with different people, different populations, different continents they tested it on, and they were able to bring that down to 3 metabolites through a ton of validation and reproducibility studies. So that is what it takes to get down to a test that we can use to monitor. But you can see it can be done. And it will be done. It just requires a lot of work. But yes, I believe that we will get to a point where we can do that.
Excellent. So I'm not seeing any more questions popping up at the moment, but again, I would encourage you, if you do have questions following this webinar, please do send them to me at Imagine, and I can always forward them to Karen for further input. I'd like to thank Dr. Madsen for her wonderful presentation today. It was extremely informative, and I hope it will be, prove useful to all of you as you engage in your research studies. So thank you to Karen. Thank you all for participating. I do want to remind you, you will be sent a link following today's webinar to give us some feedback. We encourage you to complete that. It's obviously very important for us to shape our future webinars and understand what's helpful to you in your career path. And it's also used for us to help with accreditation, so you can get CME credits for participating in this webinar. So please do fill that out. And thank you to all our sponsors that do make this Imagine Network possible, and including these webinars. So thank you, everyone, and have a great day.